Namaste everyone, I'm your Dr. Ramesh Kasarla. In this series of videos, we will explore the intricacies of female reproductive systems. Covering topics are the oogenesis, it is also known as the gametogenesis, and mitosis and meiosis. Oogenesis, in some places I pronounced as a oogenesis, please consider it as a oogenesis. Before move forward, I highly recommend you to revising our previous videos where we extensively discussed the structural components and functions of the both male and female reproductive systems. This is the fundamental knowledge you will greatly enrich your understanding of the content to come. Let's look about the oogenesis. Oogenesis is the process of egg formation in females. It takes place in the ovaries and involves in the several stages of the immature germ cells to mature eggs. Unlike sperm production, which continues throughout the life, oogenesis starts before the birth and ends after the puberty. Understanding oogenesis is crucial for the understanding the female fertility. In this overview, we will explore the stages of the oogenesis from the start to finish. Oogenesis starts with the interphase. Interphase comes before the mitosis in the cell cycle. It is the longest phase, characterized by the cell growth, metabolic activities, and DNA replications. This interphase is divided into the three stages: G1, S phase, and G2 phase, each with the specific tasks like growth, DNA synthesis, and preparation for the division. Interphase is ensure the cells are ready for the division and maintains their functionality. After completing the interphase, cells are entering into the mitosis. Mitosis, it is the process. The mother cells, it is divided into the two daughter cells with the equal number of chromosomes. It is having the different stages of phases. Let's see the first stage, prophase, which starts the mitosis. DNA gets compact into the visible chromosomes. These chromosomes are having the pairs of the sister parts connected in the middle. The cell nuclear envelope break apart and letting the spindle fibers to get close the chromosomes. Also chromosomes like cell anchors, it moves to the opposite sides from them spindle fibers to stretch out. Prophase is like the opening act for the mitosis show, getting everything ready for the big event. After completing the prophase, cells are entered into the metaphase. During this metaphase, chromosomes now fully condensed, align along the metaphase plate, an imaginary plane equidistant from the two poles of the cells. Now spindle fibers from the centromeres attach to the centromere region of the each chromosomes, ensuring that each chromatide pair will be pulled to the opposite poles of the during the anaphase. After finishing the metaphase, cells enter into the anaphase. Anaphase is characterized by the separation of the sister chromatids. In this picture, you can see the copy of the sister chromatids. The centromeres split and spindle fibers are contract and pulling the sister chromatids efforts towards the opposite poles of the cells. Once they are separated, these chromatids are considered as individual chromosomes. After formation of the individual chromosomes, now anaphase cell enter into the telophase. It is begins when separated chromosomes reach the folds of the cells. They begin to decondense to back into the chromatin and nuclear envelope starts from the surrounding of them. Meanwhile, spindle fibers are disassembled and the cell prepares for the cytokinesis. Final phase of the mitosis, it is the cytokinesis. Cytokinesis occurs during the late telophase or immediately after the telophase. It involves the physical division of the cytoplasm resulting to the formation of the two daughter cells. So, during the mitosis process, one parent cell it is divided into the two daughter cells with the equal number of the chromosomes, means 46 number of the chromosomes. But during the meiosis, that chromosome numbers will become to the half, means 23 chromosomes. Let's have detailed look about the meiosis process. During the mitosis process, female immature germ cells are producing the oogonium. Now oogonium is enter into the Meiosis. This meiosis is also having the same phases like prophase, metaphase, anaphase, telophase and cytokinesis. But during the meiosis, it is having the two phases, meiosis 1 and meiosis 2. Meiosis 1 is started before the birth and it is finished after the puberty. Meiosis 2, it is start after the semination and means egg is touched with the sperm. It will start the meiosis 2 and it will finish immediately within the few hours. Now enter into the prophase. Prophase, it is the lengthy and complex phase of the meiosis in the human oogenesis. This phase begins during the fetal development but fuses until the puberty. Means prophase 1 is started before the birth but it is in the arrested stage. During the puberty, primary oocytes, which are the diploid cells, resume the meiosis and progress through the prophase 1. Now primary oocytes are arrested state in the before birth. Now it is completed the prophase 1 by crossing over occurs between the non-sister chromatids within the tetrads and resulting in the exchange of the genetic material between the homologous chromosomes. As prophase 1 progresses, the nuclear envelope breakdown and spindle fiber 2 starts to form. Metaphase follows the prophase 1. At this stage, the tetrads line up along with the metaphase plate, a plane equidistant from the two poles of the cells. Each homologous chromosomes within the tetrad is attached to the spindle fibers and emanating from the opposite poles of the cell via centromere. The orientation of each tetrad along the metaphase plate is random and contributing to the genetic diversity in the resulting of the gametes. After finishing the metaphase 1, cells are entered into the anaphase. It begins with the suppression of the homologous chromosomes, which are the pull towards the opposite poles of the cell by the centration of the spindle fibers. Unlike mitosis, where the sister chromatids are separated in the meiosis, homologous chromosomes are separated, ensuring that each daughter cells receive the one of the chromosomes from the each homologous phase. Telophase 1 followed by the anaphase 1. During this phase, the separated homologous chromosomes reach the poles of the cell. Nuclear envelopes begins to reform around the chromosomes and chromosomes begins to decondense. Final step of the meiosis 1, it is the cytokinesis. Cytokinesis is the physical division of the cytoplasm that occurs after the telophase 1. In human oogenesis, cytokinesis 1 results the formation of the two daughter cells called as the secondary oocytes, each containing the half of number of the chromosomes as the original primary oocytes. Means parent cell is having the 46 chromosomes. Now daughter cell it is having the 23 chromosomes. One of these daughter cells retains the most cytoplasm around the organelles and it is called as the secondary oocytes, while the other daughter cells called as the first polar body. Usually it contains a very little cytoplasm and eventually degenerates it. But in some cases primary polar body also enter into the meiosis 2 and it will produce the other secondary polar bodies. After the completion of the meiosis 1, these secondary oocytes enter into the meiosis 2, which is initiated upon the fertilization by the sperm cells. Meiosis 2 results in the formation of the mature egg and second polar body it will produce. But remember, meiosis 2 it starts after the sperm cell touching to the egg. Until now, that immature germ cells are finished the mitosis and it is enter into the meiosis process 1 and it is completed the meiosis 1 and egg is ovulated. When egg is ovulated, it is transported into the fallopian tube and it is waiting for the sperms. When sperms are coming and touching to the egg, it is automatically activate the second process of the meiosis 2. During this meiosis 2, it is having the prophase 2, metaphase 2, anaphase 2, telophase 2 and cytokinesis 2 also. Let's see. During the prophase 2, the nuclear envelope break down and spindle fibers start to form, and chromosomes, each consisting of the two sister chromatids, condense to become a visible under the microscope, and then it will enter into the metaphase 2. During this metaphase 2, chromosomes, each composed of the two sister chromatids, align along to the metaphase plate, similar to the metaphase in the mitosis and meiosis process. During this process, spindle fibers from the centrosomes attach to the centromeres of the each sister chromatids, ensuring that they will pull the opposite poles during the anaphase 2. Anaphase 2 begins with the separation of the sister chromatids, which are the pulled towards to the opposite poles of the cell body by the centration of the spindle fibers. Each sister chromatids is considered as an individual chromosomes once they are separated. Unlike in meiosis 1, there are no pairing of the homologous chromosomes in the meiosis 2. Only sister chromatids are separated. Telophase 2 followed by the anaphase 2. During this phase, chromosomes reach the poles of the cells and nuclear envelopes begins to reform around of the chromosomes. These chromosomes start decondense and spindle fibers are disassembled. Final phase of the meiosis 2 is the cytokinesis 2. During this cytokinesis, the physical division of the cytoplasm that occurs after the telophase 2. In human oogenesis, cytokinesis results to formation of the 4 daughter cells, each containing the haploid chromosomes, means 23 chromosomes. Among these daughter cells, only one becomes the mature egg, while other smaller cells are called as the polar bodies. These polar bodies usually degenerate and they are not involved in the fertilization. As I told you previously, during the meiosis process, we received the one secondary oocyte and one polar body. That polar body also produced the two polar bodies during the secondary meiosis 2, and secondary oocyte, it is producing the two daughter cells. One is the polar body, another one is the ovum. This ovum is involving in the fertilization. These are the wonderful steps involved in the oogenesis. Let's once again recall the entire lesson. Before birth, immature germ cells are enter into the interphase. When it is finished the interphase, it is entering into the mitosis. Mitosis, we are having the prophase, metaphase, anaphase, telophase and cytokinesis. During this entire process, we are getting the two daughter cells with the full number of chromosomes, 46. Those are the diploid cells. After finishing this process, cells are entering into the meiosis 1. Before birth, prophase 1 is started. It is completed after the puberty. After the completing of the prophase 1, cell is entering into the metaphase 1, anaphase 1, telophase 1 and cytokinesis 1. During this cytokinesis 1 completion, we are getting the one mature secondary oocyte and one polar body. This secondary oocyte and polar body is entering into the meiosis 2 after the insemination with the sperm cells. Meiosis 2, when it is begins, that prophase 2 it is started and it is followed by the metaphase 2, anaphase 2 and telophase 2, and finally it is completed the cytokinesis 2. After completing this cytokinesis 2, we are getting the 4 daughter cells. 3 are the polar bodies, 1 is the matured ovum. This ovum is involved in the fertilization. This is the video. In our next video, we will explore the intricate process of the follicular developments, individual steps in details. For deeper understanding, visit my channel Dr. Ramesh Kasarla. Don't hesitate to share your suggestions in the comment section below. Thank you. Dhanyavad. Be healthy.